Plant parasitic nematodes cause severe damage and yield loss in major crops all over the world. Available control strategies include use of insecticides or nematicides, but these have proved detrimental to the environment. This scenario provides an opportunity for the utilization of technological advances like RNA interference to engineer resistance against these devastating parasites. In 1998, the American scientists Andrew Fire and Craig Mello published their discovery of a mechanism that can degrade mRNA from a specific gene. This mechanism, RNA interference, is activated when RNA molecules occur as double-stranded pairs in the cell. A nematode, Melodigene incognita, infects the roots of tobacco plants and reduces the ability to take up nutrients and water, thus reduces the production of tobacco. The infection can be prevented using the RNA interference technique. RNA interference helps in developing resistance in tobacco plant against nematode infection. Agrobacterium tumefaciens, a bacteria, is commonly used as a vector for the introduction of foreign genes into plants and consequent regeneration of transgenic plants. Agrobacterium has a large plasmid known as Ti plasmid. The nematode-specific genes are identified and isolated from the nematode Melodigene incognita. The Ti plasmid and nematode-specific genes are ligated by an enzyme DNA ligase. These specific genes are essential for the growth of the nematode. The recombinant DNA is then introduced into the tobacco plant in such a way that both sense and antisense mRNA is produced. Normally, when the DNA is transcribed into mRNA, only 3' 5' strand of the double helix is used. This strand is called the non-coding or antisense strand. This antisense DNA strand transcribed to form sense mRNA. Genetically modified tobacco plant contain an additional nematode-specific gene. Here in this case, both the DNA strands transcribe and form a sense mRNA and an antisense mRNA. These two RNAs are complementary to each other and form a double-stranded RNA. The specific gene which is essential for the growth of nematode transcribe and form mRNA in the nematode cells. The double-stranded RNA is then entered into cells of nematode when it infects the tobacco plant. The double-stranded RNA then binds to an enzyme complex called RISC, RNA-induced silencing complex. Here, the two strands of the double-stranded RNA separate. One floats away while the other remains attached to RISC. This strand is called as the guide strand. The guide strand binds to the mRNA through complementary base pairing, bringing the whole RISC to the mRNA. The RISC complex then cleaves and degrades the mRNA, preventing it from being translated into protein. That means it effectively silencing the gene. So the parasite cannot survive in a transgenic tobacco host plant and so prevents the plants from pest. The host-generated RNA interference has proved to be a revolutionary approach for the delivery of double-stranded RNA or small interference RNA into the feeding nematodes for the silencing of vital nematode-specific genes.